Thank you everyone for coming to this talk today. I am Ramesh Raliya, and those who don't know, I am research scientist at Washington University in St. Louis, and my research interest is use of smaller nanoscale particle for beneficial health and biomedical engineering application. Today I am going to discuss about how we can use smaller particle, nanoscale particles, particularly metal gold nanoparticle for the delivery to brain. And the intention is how we can deliver rapidly the drug delivery to the brain. So we're moving forward with the motivation from President of the United States. He is talking about that we can use a particle, or we can understand and study the particle smaller than atom, but still we are not able to understand the body organ which is called the brain. And why it is more important, because we can see that thousands of diseases are related to the brain, and these diseases require the treatments, and when we go for treatment it takes long period of time. And the reason behind that why it takes long time, because our brain has protective barrier that called the blood-brain barrier. The main function of blood-brain barrier is to protect our brain. But when we need to deliver any drug to the brain, this protective barrier causes a problem. And the problem is this protective barrier, and here is a blood, and this is our brain, and this is barrier called the blood-brain barrier. So when we put any drug, it takes long time to cross this barrier, because this is sealed barrier and it cannot cross. So how we can address that issues? So currently, to deliver the drug to the brain, there are several techniques being used by the doctors and in the medical field. So the mainly important techniques are the invasive technique, in which in case of the emergency, doctor directly use some sort of technique which can pierce your brain skull and into the brain, and they deliver the drug into the brain. But in that case, the challenge is that because our brain is control the entire body organ, and when we inject anything into the brain, there is a chance that it can dysfunction our one of the body function, and that we do not want. So in alternative, doctor use the other techniques like pharmacologic and the physiologic technique to deliver the drug into the brain. And in this technique, we can deliver the drug to the brain using without dysfunctioning or without disturbing the other body function. But the problem is it takes long time. It needs 1 to 2 months, or sometimes maybe months and months to reach the drug. Depends on that what kind of drug and what kind of drug concentration we are using. So we need a technique. Using that, we can deliver the drug very rapidly to the brain. It's a non-invasive technique, and without dysfunctioning the other body part or the brain function. So how we can do that? So we use the aerosol technique. So aerosol technique to deliver the drug. And aerosol by definition is any particle suspended in the air or the gas. So how this technology works? Let's compare with the conventional technique. What is the problem? So you can see this is a complex body network, and in the current approach, we inject any drug into the body, and this drug circulates through the entire circulatory system in the body, and then it has to cross the blood-brain barrier. So that's why it takes long period of time. But how about if we use the nasal pathway? So the nasal pathway is basically this is an oral cavity, that is the dissection of the human mouth, and here is a nose, and here eventually it will come as a lung. So in the mouth we have some olfactory nerves, and these olfactory nerves, the main function of the olfactory nerve is to sense the gaseous molecule. When we sense any kind of smell, these smells can be sensed using these olfactory nerves. And so our target is how can we use this pathway to deliver the drug into the brain. because because if there is any kind of smell, we can sense in a minute or in a second. So if we can use or mimic this pathway, we can deliver the drug in a fraction of minutes or fraction of time. So in aerosol technique, uh, because there are consequences that there is different size of particles, and when we breathe these particles go and deposit into the lung. So there is a principle based on the particle diameter, that what is your particle size, what kind of particle size is there, and based on the particle size, it determines that where this particle will go and deposit in your body. So if your particle is more than 1 micrometer, this particle will go and deposit in your lungs. So that's why the nanotechnology play an important role, that when we bring down the scale into the 5 nanometer or less than 5 nanometer, the 80% or more, all the particles get deposited in the oral cavity. So we use the same principle. We make the particle smaller. If we can make the particle smaller than 5 nanometer, we can really deliver these particles we can retain all these particles into the mouth cavity, and all these particles can sense by the olfactory nerves, and then these particles can be delivered to the brain. And if we can deliver these particles to the brain, then we can attach any kind of drug, or we can insert any kind of drug with these particles, and drug can be delivered to the brain. So to prove this hypothesis that we can deliver the smaller scale particle to the brain, here we hypothesized an experimental plan, and in this experimental plan that we first make the particle using sol gel chemistry techniques, and then we characterize this particle, that what are the properties of this particle, because we need biocompatible material which is non-toxic to the cells, and then we characterize this material using the different techniques for their morphology, for their size, shape, what is their surface charge, how is their distribution in the solvent, and how is their stability. And once we synthesize and characterize this particle, we functionalize them, because here we functionalize to track the particle, that where these particles are going. So using the microscopy technique, we can understand that where these particles are moving inside once we spray, and where these particles are moving. But eventually this functionalized technique we can mimic to functionalize with the drug. So the same technique we can, instead of the functional group, we can put some sort of drug there. Then we deliver this particle using an aerosol technique. The aerosol I described as a particle suspended in the air and gas. And then we eventually, once we delivered the particle, we took out the brain, we did the brain surgery, and then we understand that how much particle really went into the brain, and we understand the biodistribution and biointeraction. So let me start with characterization of the material when we synthesized. So here you can see that we synthesized gold nanoparticle, and these particles on the screen you can see the transmission electron microscope image, and this image is a lower scale magnification, and this is higher magnification. And the importance of this image is basically it's showing that all the particles you can see they are having the same particle size or the similar particle size. And in terms of the technology, characterization technology, there is a index, polydispersity index, that how different size of particles are present. So the polydispersity index, if index is vary from 0.001 to 1, if index is close to 1 means your particles are highly polydispersed. You have different size of particles. And if index is close to 0.001, then it shows that your particles are highly monodispersed. So here the polydispersity index of these particles shows that these particles are highly monodispersed, that having the same particle size. So whatever effect and whatever entry of these particles we will get, that will be all from the same particle size. So once we have these particles, it's really required to functionalize. Functionalize either with the particle to track, as I said earlier, with the fluorescent marker, or with the drug. So here we did functionalization with the fluorescent tagging. So here you can see that this is gold nanoparticle. Then we use a amino acid, it's called the cysteine amino acid. The cysteine amino acid has an important property which has the functional group sulfohydryl SH group. You can see at the top of the SH group, and when this SH group has a property to bind on the gold nanoparticle surface, and once it binds to the gold nanoparticle surface, we use a fluorescent dye, fluorescent isothiocyanate, and this fluorescent isothiocyanate is bind to the NH group of the cysteine amino acid, so that we have now the functionalized particle. And to characterize that whether the particle is functionalized or not, we characterize using the fluorescence microscopy, and here you can see that the shining spot is basically showing that particles were functionalized. And at the different stage, we can also visually see that in the solution, the color is changed, so that qualitatively we can say that particles were functionalized. To further confirm, we also did the absorption spectroscopy, and we see that when we functionalize the particle at different stages, like with the cysteine amino acid or the cysteine amino acid with the fluorescent tag, the peak is shifting left to right, and the characteristic peak absorption shows that particles were functionalized at the different time interval. So once we have the functionalized particle, the next task is come that deliver this particle to the through the aerosol. So suspend the particle into the air and gas so that this particle can go into the mouth cavity or to the aerial way so that particle can transport to the brain. So here you can see that experimental plan, that this is the locust sketch, and since the locust has the two antennae, they are similar to the human nose, and we choose the locust because locust olfactory nervous system architecture is pretty much similar to the human human olfactory nerve architectures. So this is an electrospray technique. Electrospray is an aerosol technique in which you can apply a high voltage to generate a monodispersed aerosol particles. So when we apply the voltage, this generates smaller particles, and these particles, along with these particles, you can see that some green color in background. So green color is basically some sort of smell. Here we use a hexanon so that locust can sense, because without sensing we cannot understand that locust is responding to these particles or not. Later I will show you that why really we use the hexanon and why it is important to use as a carrier. So we use a carrier gas to deliver these particles, and you can see that this droplet is being shrinking. So this is really here, this is a solvent because this gold particle was suspended in the water, and here is a droplet of the, and in single droplet we have a single particle. And as this droplet travels through this tube, this droplet gets evaporated and eventually you have a single gold particle which is functionalized. And these particles will deliver to the antenna. So here you should not assume that we actually pump everything to the locust mouth or brain. No. In the experimental plan, we only expose the locust antenna. The rest body part were not exposed to the bone. And at the same time, we also use some. We also measure the brain electrophysiology using the projection neuron. So we use the projection. We did the projection neuron recording for the electrophysiology, that how the brain functions. This barcode is basically showing that when any gas or particle interact with the olfactory nerves, then nerve gives some sort of signal to the brain that hey, some sort of smell is outside, or some sort of ligand molecule is outside, so that brain gives signal to the body that hey, this kind of function we have to perform. So this is called the brain electrophysiology. So once we expose this particle, now comes that how we quantify and understand that where this particle went into the brain. So once we expose, we did a brain surgery. We opened the brain, the skull of the locust, and then we take out this locust brain to the one. And once we take out, then we did a fluorescence microscopy study to look at that within the brain where actually these particles are using the fluorescence microscopy. And once we understand that these particles were in the brain, but we are not able to say that how much particles were there. And why it is important? It is important because when we use the pharmacological drug, then we need to quantify that how much drug we are delivering to the body. Because always drug is measured in terms of like per kg body weight. Like for the child we are giving a different dose concentration, for adult we are giving different dose concentration. So that's why it is important. So then in this progression we use some acids. So you can see some dot that basically shows that we use some acid to digest the brain in the acid to quantify. So once we digest the brain, digest means basically we dissolve the brain. We use an inductive coupled plasma mass spectroscopy, and this inductive coupled plasma mass spectroscopy technique allow us to quantify that how much number of particle and how much amount of particle we deliver to the locust, so that we can quantify the amount of drug we are giving to the brain. So here are the result of the biodistribution. So this is an experimental sketch that we delivered to the gold aerosol, the gold nanoparticle aerosol, and this is the brain we took out. Once we take out this brain, we did the fluorescence microscopy of the antenna on the brain. Antenna one we exposed, and the brain which was not exposed. And both the figure you can see that there is some sort of fluorescence. So it represents that, and it suggests that there are particles were transported to the brain. In the antenna, one thing is to be noted that some sensilla, some spikes are more glowing than the others. It represents that some sensilla has receptors to receive these particles or receive this kind of signals. And in the brain, you can see here that the entire brain is glowing. So, but our target is to focus on a particular site in the brain that we are targeting in the next phase of the study. But to see that once these particles go into the brain, whether these particles remain in nanoparticle form or not, or these particles get metabolized inside the system. So we did the transmission electron microscopy, and here in the green circles you can see some sort of black dots. In these black dots are basically the gold nanoparticles. Now you can raise a question that these are not the gold, these may be some lipid body or some other things in the brain. How can we confirm that these are the gold particles? So there is a technique that called this technique coupled with electron microscopy that called energy dispersive X-ray spectroscopy, EDX technique, that basically quantify the elementally that whether these particles were gold or not. But here I'm not showing that result. Once we qualitatively understand using the fluorescence microscopy and transmission electron microscopy that these particles were in the brain, then we quantified using the ICP mass spectroscopy that how much these particles were accumulated in the brain. So here you can see that this is an unexposed antenna. We considered as a control. So unexposed antenna means the locust has two antenna. One antenna we exposed, another antenna we did not expose. So these are the results from unexposed antenna. And here is the result of the exposed antenna, the antenna one we exposed. After the exposure, we terminated the experiment. We cut both the antenna and we did the ICPMS. And then eventually we did the ICPMS of the brain. So the trend basically is showing that the accumulation of nanoparticle in the brain is increasing with the time from 1 hour to 6 hours. So the question, it can make sense that particles were accumulating with the time. But the question arises that why the gold in the exposed antenna? Unexposed antenna. It can be attributed because at least 1 hour exposure of unexposed and unexposed antenna gold is equal. So the question is how this gold is came into the unexposed antenna. So initially we thought that there is a kind of contamination, but later we performed several experiments and we did the background analysis using the material, that what kind of material we are feeding to the locust. So we find that we found some sort of gold content in the material we are providing to the locust. So all the controlled locust, even we are not exposing any kind of gold, we find the gold. So that gold is coming from the material we provide as a food material to the locust, from the soil, from the grasses, or the oat we are providing while spawning these locust colony. And finally, we know that these particles were reaching to the brain, and in this X amount of particles were reaching to the brain. Then question that how these particles behave inside the brain, because our aim was not to disrupt the brain function. So here we use projection neuron recording using the multi-channel electrode, and these barcodes, these barcodes basically showing that how the neuron functions, and the number of bars here showing that how many times neuron spike when neuron get any signal. So here you can see that when we only expose the hexanol, because hexanol is a particular kind of order, and this order is studied well in the locust study, that when you expose X amount of hexanol, then how much spike you will get. So that's why we use the hexanol. So you can see here the response with the hexanol without gold nanoparticle, and when we use the gold nanoparticle, you can see there is a similar kind of response. So it suggests that gold nanoparticle did not interfere with the other brain function or the brain spikes. And when we quantify the number of spikes in terms of the firing rate, you can see here we did the statistical analysis, and it shows that there is a non-significant difference even when you don't expose the gold nanoparticle, or at the beginning of the gold nanoparticle exposure, or after the 2 hours of the experiment. We did not find any significant change. So it suggests that the nanoparticle we exposed to the locust was a biocompatible particle. So, so far I showed that how we made these biomaterials which can be used, which can be successfully functionalized and used to deliver this particle to the brain using the aerosol technique. And we detect this particle, their biodistribution and the biointeraction using the electrophysiological recording, and quantified using the mass spectroscopy technique. So let me conclude the talk. So here I mentioned that how we use Electrospray technique to deliver these particles, and this technique allows us to deliver a single particle at a time to the locust brain. And then we quantified this particle using the ICP-MS, and qualitatively we checked using the fluorescence microscopy and electron microscopy. And all that suggests that these particles are transporting to the brain using the non-invasive technique. And you have seen that besides the surgery, we did not pierce the brain. So it's a completely non-invasive technique. And you also seen in the ICP-MS result that these particles reaching to the brain within 1 hour. So in a short period of time, we can deliver the drug to the brain. And the electrophysiology response with reference to the gold nanoparticle, it shows that gold nanoparticle did not interfere with the brain function. So they are really biocompatible materials. So, and these particles can be used because these particles are biocompatible material and they are not interfering to the brain. So these particles can be used as a drug or drug carrier with no time, and using a non-invasive approach, we can deliver this particle to the brain. So how we can use, or what will be the eventual goal of of such study or these kind of studies. So this study open a path for delivering the drug to not only to the brain. The one example I showed the brain, but not only to the brain. We can future we can also target to the other body parts, so that you can simply spray the drug on the nose, and the drug can reach to the body part, and it do not require any kind of expertise, because if you need an injection, you need to go to the hospital. But if there is a technique to deliver the drug using the spray, even a kid can do by himself or herself within no time. So with that, I would like to thank my research collaborators at Washington University and the animal I sacrificed in my study. And with that, I would like to take your questions. Thank you so much.